You have already learned about the amino sugars. Similarly, the second important portion of the GAGs are the acidic sugars. The two acidic sugars which are used in the formation of GAGs are the glucuronic acid and hydronic acid. The glucuronic acid is formed from glucose. Glucose, of course, you get from the diet. The glucose is converted into UDP glucose, and the UDP glucose then under the action of UDP glucose dehydrogenase give rise to the formation of UDP glucuronic acid. So, as you know that the GAGs are not only composed of the amino sugars, but also of the acidic sugars. The two important acidic sugar moieties are the glucuronic acid and hydronic acid. The glucuronic acid comes from your diet and also from the metabolism or degradation of GAGs. The glucuronic acid is formed because I have just told you that it comes from diet because it is formed from glucose. The glucose was changed into the UDP glucose. This UDP glucose molecules under the action of glucose dehydrogenase or UDP glucose dehydrogenase is converted into UDP glucuronic acid. Once you have the UDP glucuronic acid, it must not be difficult for you to know what it will do next. Yes, it will then become the part of the elongated chain of the GAGs. Why? Because we have already learned that once a molecule acquires UDP, it's become energetic and it can easily then be involved in the reaction, so as to become the part of the elongated chain. The UDP galactose, once it's formed, it not only UDP glucuronic acid, once it is formed, it is not only doesn't only become the part of what we say the GAGs, but it is also involved in the formation of many other molecules which are polar in nature, for example, in the formation of steroids. Similarly, in addition to the glucuronic acid, there is also hydronic acid, the second sugar moiety which is acidic in nature. This also is incorporated into the GAGs formation by getting incorporated into the elongated chain. It facilitates the epimerization of D2L sugar. So, D2L sugar epimerization occurs under the enzyme uranosyl 5 epimerase. So, keep in mind that though many of the reactions of the biosynthesis or even degradation of GAGs or proteoglycans are very similar to the biosynthesis of glycogen, the major difference is that biosynthesis of glycogen takes place in rough endoplasmic reticulum. From there it is transported to Golgi bodies and then towards the outside. So, this is one thing you should always keep in mind. After the glycosylation, the Golgi bodies is endoted towards either the cytoplasm or towards the ECM. Now, a little bit I want your focus now. We are going to learn how this compound is formed. Again, if you revise, if you memorize, if you then freshen your memories, you should have in your brain the very shape, very figure of this molecule, the proteoglycan, a protein molecule, a protein core protein, and then that core protein is linked with trisaccharide, I mean 3 sugar molecules, and then these 3 sugar molecules in turn are linked with the repeating units of the GAGs. So, this is you should keep in mind. First of all, we must know that how these 3 sugar molecules which act as the linker molecules, they become part of the proteoglycan in the synthesis of the proteoglycan. So, here you know this is the core protein for in the rough endoplasmic reticulum. This core protein first of all gets the sugar moieties which will act as the linker molecules. These, for instance, you get the linker molecules which are composed of xylose and 2 galactose moieties, and then after xylose and 2 galactose moieties are formed, then they are linked with the serine residue of the protein, and then in this way now the platforms is formed for the incorporation of individual repeating units of the GAGs. So, once you have the linker molecules and then a new molecule from the for the formation of or new hexose or sugar molecules added, and once the new sugar molecules added, then what will happen that I mean usually keep in mind the number of units disaccharide units of the GAGs which it goes to around 150 to 200. This is something you should know that it is not a small molecule, it is a large molecule. Then once the final residue has been added, the final residue of the carbohydrate moiety of the proteoglycan has been added, then it undergoes the process of sulfonation. This sulfonation is applied through the activity of a compound known as PAP. PAP provides the sulfur moiety. This is how this is you know you have let us say the carbohydrate moiety of the proteoglycan, and it gets sulfonated on the very terminal residue, very terminal sugar residue, and of course, the sulfur transferase enzyme is involved in this reaction. This enzyme is not sulfur transferase is not only involved in the synthesis of proteoglycans, but is also involved in many other major molecules like glycosphingolipid synthesis also. In that also the sulfonation occurs through sulfur transferases.